pure water cannot conduct electricity. It's got to have salts dissolved in it, like it gets out of the ground when it comes in your house. That's why the salts are in here, to carry the electricity through this whole thing. Okay? This soap in there actually holds DNA together, so it doesn't break apart as it's being tugged through here. So, when you get back to your station, when I'm done talking, you're going to have goggles from over here. There's gloves over here. I'm guessing most of you use small. I did bring some mediums if you need them. And I'm going to go around with this tub of all the other gels you're going to need. Okay? So, you're going to get to your station. You're going to set the chamber out. You're going to put the gel in there with your gloved hands on. The oils from your skin, if you get on the edge of that, it'll actually monkey up the whole run. Instead of the DNA going in straight lines, it'll bend and twist and stuff. So you got to make sure you do this with clean hands. Once you do that, one of you or both partners in the group, you're going to put your hand up. I'm going to come over and flood the chamber. Go on one side, go the other side, and let it go right across the top just a little bit. Now, once it's in there, comes the tricky part. You have to put your 6 DNA samples in the 6 holes in there. Can you even see those holes?
No.
You can if you move around enough. You got to get to the place. I got to do this. You won't be able to see it from back there. You got to get to the right place where you put them. Now, these are labeled 1, 2, 3, 4, 5, 6. I suggest you start at the top and go 1, 2, 3, 4, 5, 6, so we all end up with the same thing to look at them. Okay? Here's what you do. And you already practiced this part. Put it in there. Draw it up. Find the first opening and put it right down in there. There's a little bit left. You can just go up and back on the sides, kind of dry that off. Now, if we were using real DNA, we'd have to throw these out every time. But for safety's sake, like I already told you about, we're going to use these fake DNA, and we can get away with using the same tip over and over. If by chance one of these you have is dry or almost empty, bring it up. I have like 100 more with me. Sometimes if the cap didn't get shut tight from the last class, they dry out. That's not a big deal. Okay. Because most of you are in groups of 2, maybe one person does 3 of them, the other does the other 3. Of course, a group of 3, 2 and 2, whatever, practice this. Or I should say divide it up a little bit. Can you give me another tip out of that bag? That tip seems to be losing some a little bit. Thank you. There we go. Now, like I said, I've been doing this for a long, long time. Don't try and rush through it like I am. And also, as I said, don't worry about how many steps I'm giving you, because they're all outlined right on that piece of paper at your table. Okay? And of course, I'm going to be here if you have a question. Once you start putting these in, don't move that box. Don't bump the table or anything, because the little waves will wash them all back and forth and can mess up your whole experiment. So you're going to be very careful not to hit the table or anything. Now, the rest of this is really, really complicated, so pay attention, okay? Once it's in there, once they're in there, you put the cover on. And you bring your battery pack over. Now, they can only fit on one way. See how this one's like mushroomed open? Yeah. That goes on the round end. And this one's mushroomed open, so you got to put the round end here in. So you put the cover on, you put the battery pack, and you're done. Really complicated, right?
Yeah.
I was kidding. Now, once everybody at your table has this sealed, then you can take the glasses off, and the gloves come off one way. You grab the cuffs, turn them inside out, and just set them on the table. Later on, I'll tell you how to flip them backwards so you can use them again. So what I need you folks to do is to get to this point. Okay? Why don't we do this?
Okay.
Have a question?
No.
Oh, that's right. I told you to put your hand up, didn't I?
Okay. You look like a You look like a Yes, I got it perfectly. That's awesome.
Nice. Let me grab another bottle.
I can't see number 1, but I can see. Oh, you're sure? That looks really nice. Yeah. Wait, is that?
Yeah, it's 3.
No, look how much we have.
Oh, yeah, there's enough.
Oh no.
See, you need to have it like
being messed up.
Not all the way down, because okay.
Right there.
No, there's not enough in there.
Oh, that's cool.
See?
We're gonna have to sort
it out on the side.
Yeah, let's have to sort it out.
The pieces of jelly, they're underneath it, brother.
Orange, whatever.
Yes. You got plenty in there. Then you already stuck the side of your kitchen.
Very careful.
There you go.
There's just...
No, it's not coming out.
Yeah, folks, if you know there's liquid in the tube, but it's not picking up, that's a good sign that that's
The cap in the water.
It was No. Oh.
It's in there.
No. There's the one it's in there. But you don't touch the bottom of it. It'll sway out.
You're supposed to hold it, then take it out, dude.
Oh, you just put it inside. Stop shaking the table. Okay, cheers. Can you feel it?
Wait, I did one.
Basically, we took this thing and we put this special yellow thing on it, and we had these things.
Okay.
What they are is they're gels that we put in here that make the chemicals show up. Because
You would
real life,
stick this thing in there, and then you would stick it in those holes that have the things in there, one of those things. And then after you're done, you close that, and then you close the top.
Okay.
And then you take the batteries, and you have to hook them
Put the
up like that.
mushroom side with the circle
Yeah.
and the circle with the mushroom.
Yeah, and then it's like putting electricity in it because there's salt water and soap in there.
Yeah, there's salt water and soap in there. And what it was found on accident somehow.
Can somebody take that remote
So
and
this
turn that
is
thing on?
technically DNA fingerprints.
So this is the completed one. You want to tell me how this works?
Sure.
You had to put all the fluids into the little tiny holes in there, and then Miss Brisk squirts the water into the
Like I'm half checked.
chamber, and it floods it, and then you get your battery pack and you hook up these two wires, the red one to this side and the black one to this side, and make the circle one go into like the flower shaped one, and the circle, and then the flower shaped one go in the circle one.
Very nice. Thank you.
Are actually water molecules being split in half by electricity. It's called electrolysis.
Look at all
Those
those
bubbles are pure
bubbles.
oxygen and
Look
pure
at on the other
hydrogen.
side. I know.
So, would it be very smart to have a Bunsen burner going on the table with these? Oxygen and hydrogen both explode. Yeah. These little chambers don't make enough that we have to worry about it. But those are actually explosive gases that are being made in there, believe it or not. Now, the other thing is, you should be seeing them sort out, separate out by color, but that also means the sizes are different. It's sorting this whole thing by size, okay? If you want to, it doesn't matter which way we really do this, but I think what I'm going to go ahead and do right now is explain that chamber in more detail. Then when you open it up and look at your results, you'll have a little better understanding of it, okay? So there's a new thing here. By charge. Some of them are positive, some are negative. What do you think is going to be drawn towards the positive pole? Think of magnets.
The negative.
The negative, the opposite. And the negative pole, which is the black, is going to attract the positive. So these chambers sort by size and by charge. When you break DNA up into fragments or pieces like this, very often they have charges on the end. So it's sorting in two ways. Now, if you go on to become a forensic scientist, you're going to find something out the minute you go into your first lab. The chambers they use, like down in Albany in that forensic science lab, they're not 3 inches by 5 inches. They're more like 2 feet by 3 feet. The gels are huge. Okay? Because they have to sort out. You're going to end up with 2 or 3 fragments, different colors. Real human DNA has 2 or 3 thousand fragments. So when they run this gel over maybe a day or two and they look at it, they're going to have hundreds and hundreds of bands all over this thing. That's what they're going to be examining. Again, they have controls in there, but the other ones would be a suspect in a crime scene. That's what your number 4s and 5s are. A suspect sample and a crime scene sample. When we tear these down, you have to figure out if the two match. If they match, your DNA puts somebody in a crime scene. If they don't match, then you can say they don't match. Does it mean they weren't at the crime scene?
Nope.
No, it just means they didn't leave any DNA there. Okay, so it goes kind of flip-flops back and forth. Okay, with me so far? Or up to this point, maybe I should say? Now, let's talk about one more thing about this DNA, and then we will open these chambers up. The DNA is specific to one person. I keep saying that. But there's a little problem with what I'm saying. What two possibilities could give us people with the same DNA? If you clone somebody. To the best of my knowledge, no human being has ever been cloned. And I have never been able to come up with any reason why one ever should be cloned. Let me explain something to you. I'll use my family, my dad, as an example. People have a wrong idea about cloning, and you usually get that wrong idea from movies that fake it all. Okay? I lost my dad in a car accident about 20 years ago. Okay? Every day I think of him. I'd love to have my dad back. Okay? But let's say, like, if I found an old comb of his and found some hair cells and I cloned him, would I get a 6'3", 250-pound guy standing next to me?
No.
What am I going to get? A baby. Is that my dad? When you clone something, you start a new organism with the same DNA. You do not remake the person. Doesn't happen that way. What made my dad was his 9 brothers and sisters, living on the farm in Schuylerville, being in World War II, running General Foods Corporation, being a Wilton County supervisor, everything from his past. Does any of that exist anymore? No. Can you clone somebody?
Yes.
You make another human with the same DNA, it's not the same person. Okay? People really need to understand that. How are you going to use this DNA? What are applications of it? Okay, what could you find at a crime scene that would have DNA on it? What? Blood?
A desk or something that they touch?
They'd have to, maybe just a light touch. It's a real long shot. But if they scrape their arm on it or something, left some skin cells, certainly. Hair, if it's ripped out by the roots. Once you get the gloves back on, the gloves and glasses back on, then you have to take this chamber apart, and that's what I want to explain to you. Okay. I'm getting there. What's up?
I have a sample of DNA.
Yeah. Okay. Now, everybody listen carefully. This is simple as long as you pay attention. You're paying attention over there. Gwen, pay attention. Okay. First thing you're going to do once you have the gloves and glasses on is to disconnect the battery. If you just grab the wires and pull them, you could break them. I've had them broken in the past. So please grab it by the clasp up there.
What are you doing now?
You're gonna take it out. Watch out.
You're supposed to
Taking it apart?
You leave.
Wait, we bring these up to the front.
Leave that.
No,
I
I never
think you
said
put
that.
it right
When
there.
you don't
Put it right
listen.
there.
Everything but that.
Mr. Bestwood did.
I'm coming over your shoulder here a bit.
Oh, you pour it back in?
Yeah,
You reuse it? Oh, cool.
Yeah. And the gels you just used, we remelt that. On this experiment before.
Nope.
Okay. Do you usually do something perfect the very first time you do it?
No.
So, is the result as important as the lab we just did?
Nope.
No. So if it comes out a little goofy, if part of it didn't work, that's not the point here. Don't even worry about that. Okay? The point is understanding and doing the experiment itself. If you did this 2 or 3 times, then I guarantee you'd get it perfect every time. Okay? But just on the very first time, don't beat yourself up if it came out a little goofy.
This is a good way to end a class, isn't it, guys?